Today's video is on the really fascinating topic of prostate cancer and how within a matter of weeks we've expanded our knowledge base tremendously insofar as genomic markers that denote the risk of prostate cancer, not just whether or not prostate cancer is in the card, but also whether or not it would be a particularly aggressive or non-aggressive marker. On January 15th, the New England Journal published a study from Zhang and colleagues that looked at 5 SNPs in prostate cancer. 3 were in 8q24 independent blocks, that is, linkage disequilibrium bins that were not the same. So 3 independent markers, not in a gene, but actually in an intergenic desert zone of the genome, known to be quite interesting with its association with multiple types of cancer, solid tumors. The other 2 markers were on chromosome 17. One of them was actually shown to subsequently have a association with protection from diabetes, yet an increased risk of prostate cancer. Quite an interesting observation. So the Bowman-Gray group in Wake Forest took these 5 markers and showed quite remarkably that as you would increase the number of SNPs that were present, the risk variance, that you could get to risks that were almost tenfold by aggregating the 5 SNPs information. And in fact, on the front page of the New York Times, it was even announced that a $300 test was being soon to launch based on this work by the group of investigators at Wake Forest. So we thought, well, the field had actually matured quite a bit, and then just recently, in fact about mid-February 2008, there were 3 major genome-wide association study reports in Nature Genetics, and they have taken this field considerably further. Firstly, in these 3 studies, one emanating out of the UK and Australia, another coming from the National Cancer Institute with many different collaborating centers, both in the US and in Europe, and the third from Decode Genetics from Iceland with multiple cohorts. All 3 studies collectively have dug deeper for SNPs associated with prostate cancer in large populations. And what was found with replication cohorts in each of these studies is that not only were the 8q24 variants and the 2 on 17 confirmed in other studies, so these 5 SNPs were indeed validated by the new study, but many other new genomic markers were identified. One, which was quite interesting, was a marker that was in which the variant was just a couple of bases away from the promoter of a gene which is actually found in semen. So this, of course, would be one that, from a hypothesis-free work, is quite striking. Another is actually involved in the production of PSA. So this is interesting that out of such hypothesis-free research, that prostate-specific genomic markers have been identified. But collectively now, we have more than 12 new markers that have been validated in these studies, extending on chromosomes 2, on the X chromosome, on chromosomes 10, 11, and so on. And so no one has yet aggregated all this information, but it does appear that there is considerably more genomic variant definition of common variants with risk that could take that risk considerably higher than the 5 SNPs with a near tenfold risk when they were culled together. Now, there are some interesting points to also bring out from these 3 new studies in Nature Genetics. These reports come in the wake of previous genome-wide association. Why did they not show these variants earlier? Well, some of these studies, interestingly, looked at a very high-risk heritable population, such as prostate cancer in young men. And this, of course, would enhance the aggressiveness of the cancer, as well as the likelihood for finding heritability. Another study decreased the threshold remarkably for how low it would go on the SNP list from the initial genome-wide association down to 25,000, and found that, interestingly, some of those SNPs, a couple, JAZ1 in particular, and another one that I've already mentioned, actually came out very strongly when subject to the confirmation population testing, genotyping. So all in all, we have differences in the types of population tested, differences in the threshold by which the genotyping was assessed, and we have now, for the first time, lots of new information about markers that track with aggressive prostate cancer rather than any prostate cancer. More work needs to be done, particularly that aggressive prostate cancer is much lower incidence and not so much statistical power in detecting it. But overall, this has been a big new outpouring of information from intercontinental work that's been quite extensive and builds on already what was in January 2008 a significant body of knowledge. So hopefully in the times ahead we'll start to see a prostate cancer multiplex panel, which would not only predict risk for common variants, but also give us a lot more information with regard to whether the tumor is likely to be aggressive, and ultimately even response to therapy, since there looks like there may be a response to reducing prostate cancer with finasteride, but it isn't clear whether or not that drug, which can also increase the risk of aggressive cancer, would be ready. This is a type of ideal assessment study to see whether drugs that reduce the likelihood of prostate cancer are specific for certain genomic markers. So I'd be really interested in your thoughts regarding this jump in the knowledge of common variants for risk of susceptibility for prostate cancer, and hope that you will log on the site with your comments. Thanks very much.